of the most complex events that happen within the eukaryotic cell is translational initiation, and that will be the topic of this specific video of the Molecular Cell Biology YouTube channel. So, translation is a process that requires several different players. Among them, it's important to keep in mind that ribosomes will be absolutely critical because ribosomes are the actual machines that are in charge of doing the process itself. So ribosomes are composed of two different subunits. You have the large ribosome subunit and the small ribosome subunit. So that's going to be a critical component in the process, ribosomes. The other essential critical part in the process of translation will be the messenger RNA, and for a typical eukaryotic messenger RNA to be properly set up or prepared for translation, it needs to have a cap structure at the 5' end of the messenger RNA, and at the 3' end it has to have a poly-A tail, which typically consists of anywhere between 200 and 250 A's that are added at the 3' end of the transcript. So that's the other component, the one that will be used as the template. And finally, there will be two additional important players in the process of translation. They will be the tRNA molecules that are the ones that will be bringing the amino acids to the ribosome. So those tRNAs need to be charged, they need to be loaded with their corresponding amino acid, which will be added at the 3' end of that molecule. So at the 3' end, you have to have your amino acid in that tRNA. And the final set of players that are going to be absolutely essential in the process of translation are the translation factors. So there are different types of translation factors. They are the initiation factors, there are the elongation factors, and then there are the release factors. So three different types of factors that are critical for the process. Now, since we are focused on what happens in eukaryotic cells, all of these factors will be referred to as E. E for eukaryotic. So eukaryotic initiation factors, that's the first group of factors, the ones that are involved in initiation. Then the EEFs, the elongation factors, eukaryotic elongation factors, and then the ERFs, the eukaryotic release factors. All right, so all of those are going to be essential components in the process of translation. So, however, we're not really going to be talking in this video about the elongation step nor about the release stage. Our full attention in this video will be focused only on the initiation stage of translation. So the only ones that we will care about in this video will be the initiation factors. These other two are not going to be taken into consideration, so let's just eliminate them from there. All right, so let's get started with the process. So translation will initiate by recruiting some factors in the small ribosomal subunit. That's the first stage that needs to happen. And for that to be possible, you need to first dissociate the large ribosomal subunit from the small ribosomal subunit. And that dissociation is mediated by some of these initiation factors. So there is a factor known as eukaryotic initiation factor 3, and I'm just going to put the number 3 in there, that will bind to the small ribosomal subunit, and that binding by itself will be sufficient to trigger the separation of these two subunits. Now, in order to keep it open though, to keep it from coming together again, there is another factor that will be binding to the large ribosomal subunit, and that is a factor known as factor 6. So I'm going to put factor 6 here in the large ribosomal subunit, just as a way to keep tabs on what is happening. Factor 6, remember, will keep the large ribosomal subunit from ever collapsing again with the small ribosomal subunit before it is the appropriate time to do so. And that's something that will be very tightly regulated later on during translation. So, all right, so now we have our small ribosomal subunit freely working in the cytosol. All of these are cytosolic events, keep that in mind. All right, so now we need to actually load a few additional factors in the small ribosomal subunit. So for simplicity, to make this a little more obvious, I'm going to now increase the size of the small ribosomal subunit. Let's make it substantially larger. And now, actually, I think we need to also increase the size of eukaryotic initiation factor 3. All right, and now keep in mind that within the small ribosomal subunit, you have 3 different locations. You have the exit site. They are like crevices that are present in both the small ribosomal subunit as well as in the large ribosomal subunit. So you have the E site, you have the P site, and you also have the A site. So the A site right here is the amino acid site. That site is where tRNAs will typically be brought into the ribosome. The next location is the P site. The P site is the peptidyl site, which is where the peptide bonds will be created within the ribosome. And the E site is the exit site, the site where tRNAs will be allowed to exit from the ribosome. So those 3 locations are critical. They are present in both the small and the large ribosomal subunits. Keep that in mind as well. All right, so there are a few factors that need to be recruited to the small ribosomal subunit to start the whole process. And the most important factor that needs to be recruited is the tRNA. So the tRNA needs to come into the small ribosomal subunit, and the tRNA that is typically used during the process of eukaryotic initiation, translational initiation, is the one that carries methionine. So it's the methionine tRNA. Let me write that down here. Methionine tRNA. That's the first tRNA that will be brought into the small ribosomal subunit. Now I'm going to shrink this one so that we can actually work with it in a more handy manner. And now I'm going to be writing another factor that will be absolutely essential to allow for the recruitment of that tRNA, because the tRNA by itself cannot be brought directly into the small ribosomal subunit. It needs a carrier, a factor that will actually bring it and allow it to associate with the small ribosomal subunit. And that factor is eukaryotic initiation factor 2. And factor 2 is a GTPase, which means in order to be active, it needs to be associated with GTP. So we're going to draw the GTP as a circle that is associated to factor 2. And, okay, so let's actually emphasize this. And this is GTP, so since it is GTP, we are going to put a T to indicate that it is the GTP. So remember, when you see the T, that indicates GTP. So it's a guanine triphosphate nucleotide that is bound to eukaryotic initiation factor 2. Now, in the presence of the GTP molecule associated to EIF2, EIF2 is able to then interact with the tRNA that carries the methionine amino acid. So this is absolutely essential for the formation of the first component, the pre-initiation complex. And that is going to be happening all in the small ribosomal subunit, the first pre-initiation complex. So now, in order for EIF2 to be recruited, first you need a few additional factors. So you need factor 1A, which is going to associate in the E site of the small ribosomal subunit. And then you need another factor, factor 5. Now these 2 factors are being recruited by factor 3. So factor 3 is playing a key role in recruiting those additional factors. Let's give it a little bit of color so that it becomes a little more easier to keep tabs on who's doing what. Let's give also some color to the small ribosomal subunit. And now let's give some color to those additional factors that we had put in place already. So factor 5, I don't know if that's a good choice of colors. Let's actually change the colors of the factors themselves. So factor 5, and factor 1A right here. All right, so now, with those factors in place, then we can actually proceed with the recruitment of factor 2, which is the one that is carrying the tRNA. So let's see, we're giving a little bit of color to this. And now let's do the recruitment. So here we go. We are bringing all of this together in here. Now keep in mind that the tRNA will be in the P site. So that's exactly where you're going to be having all of this. I'm going to put it like this. Keep in mind the tRNA, you know what, let's just correct this so that it actually looks a little more logical. Otherwise you're going to have issues understanding the process itself. So I'm going to put factor 2 here, a little in juxtaposition with the tRNA. And now I'm going to be bringing all of that into the P site. So that makes a little more sense right there. So the tRNA is right there in the P site. All right. So now that we have that, there is another factor that will be occupying the A site, and that factor is factor 1. So let's put it in here. So eukaryotic initiation factor 1 will be located right there. And let's also give it a little bit of color. There we go. Oh, that was not too good. Better. All right, so this complex that I have drawn right there is what needs to be present in the small ribosomal subunit so that it can then associate to the messenger RNA that will be used as a template to initiate the process of translation. Now, this complex that I have drawn here is what we refer to as the 43S complex, pre-initiation complex. All right, so that pre-initiation complex is like one branch of the events that are required to allow for translation to get started. These events, as you can clearly see here, are happening completely independently from any event that is happening at the level of the messenger RNA. So this is one branch of what is happening here. The other branch will be happening in the messenger RNA. So the messenger RNA that will be used as our template for translation needs to also be activated somehow. And the key component in the messenger RNA that will be absolutely essential for the activation so that you can start recruiting all the factors that are required for translation is the cap. So that structure that I'm drawing right there at the beginning of the messenger RNA molecule, so this is our messenger RNA molecule that is being used as the template. At the very end, that structure is the cap. The one that I drew right here, that's the cap. That cap structure is going to be absolutely critical in the recruitment of all the initiation factors that then will allow for the 43S pre-initiation complex to then be brought into the messenger RNA to start the scanning process. All right, so in order to recruit the 43S pre-initiation complex, then you need to have a few factors, as you might have guessed by now, associating to the cap. And the first factor that needs to associate right there is the so-called cap binding protein. The cap binding protein is the protein that will bind to the cap. And that protein, by the way, all of these factors that will be binding to the messenger RNA, they all are members of the eukaryotic initiation factors 4 family of factors. So the first one, the cap binding protein, is 4E. So I'm going to be using only the letter initials to indicate the identity of the factors that are being recruited. So the first one is factor 4E, which is also known as the cap binding protein. So eukaryotic initiation factor 4E is also our cap binding protein. All right, now 4E will be recognized by another factor of the same family, so the 4. Well, I'm calling them a family even though they are not necessarily related proteins. It's just that they all receive the same nomenclature. They are all eukaryotic initiation factor for something. All right, so the next one is a factor known as 4G. So the next factor to come here into play is going to be a factor that associates to the cap binding protein, and that's factor 4G. So eukaryotic initiation factor 4G is the one that will then be recruiting the other factors that are required to start the process of scanning. So G will be mediating a lot of other protein-protein interactions, and because of that, I like to refer to factor 4G as the grouping factor, the one that groups all the other factors together so that they can then play the role that they play in translational initiation altogether. So I'm just going to call it the grouping. And again, this is very artificial, but this is so that it makes more sense to you. The grouping factor, the one that groups all the other factors. All right, I'm going to decrease the size here because I just realized that the location is very odd of all of these drawings. So I'm going to have to move them a little. There we go. So now I have squeeze them toward the other side so that we can work a little bit better. And the final factors that belong to this group of the 4 factors are 2 additional members of the same group that are going to be playing a very important function during translational initiation, and those are factors 4A and 4B. So eukaryotic initiation factor 4A and eukaryotic initiation factor 4B. Now, these 2, actually 4A specifically, is a helicase. But the helicase activity of 4B depends on the presence of 4B, which facilitates its helicase activity. So together, 4A and 4B provide the role of a helicase. This is an essential function during the initial scanning of the messenger RNA, because in your messenger RNA structure, this is something that is potentially likely to happen. You may have secondary structures in your messenger RNA, and I'm just making an example of what a secondary structure will look like. Now I'm going to delete this so that we have a good secondary structure in there. So if we have that structure in there, it's because we have some hydrogen bonds that are happening between the complementary sequences that you have in the messenger RNA. So this will be a stem loop. That's stem loop structure. And if you imagine the scanning process as grabbing the messenger RNA and then allowing the small ribosome subunit to move along the template, and that's something that we will be doing in just a second, in order for it to be able to scan the whole section, you need to eliminate secondary structures. If you have any of these secondary structures, then the ribosomal structure that is scanning the sequence, like trying to read through the messenger RNA, searching for the start codon, that scanning process is going to be halted and completely prevented if you have this type of secondary structures. So for the scanning process to take place normally, you need to eliminate secondary structures, and because of that, you need the helicase activity of eukaryotic initiation factors 4A and 4B, which are our helicases in the process of scanning. So, all right, the next thing that we need is then to recruit the 43S pre-initiation complex to your messenger RNA. And that process of bringing the 43S, so let's actually do something here. Let's keep it separate for the time being. Let's make a copy of this. All right, so we have those 2 separate, but now in the next event, let's now make a copy of this as well. And now let's do a paste, but let's make it slightly smaller so then you can play with this a little better. And let's do the same here. Let's shrink it. All right. So now we have a shrunk image of them. So the next stage then will be linking these two components. We're going to be bringing this complex in close proximity to the messenger RNA. And this is going to be possible because factor 4G has the ability to bind to factor 3. So these two will be able to interact with each other, and that interaction is going to then allow the deposition of the 43S pre-initiation complex onto the messenger RNA. So again, the interaction between factor 4G and factor 3 will allow these two complexes to then be brought together. So that interaction then will bring the 43S pre-initiation complex. 43 is going to be bringing small Rosas unit onto the messenger RNA. And the next thing that will happen then will be, once you have formed this complex, and by the way, I'm going to keep it separate here so that we can give it a name. When you have the two of them forming a complex, that will be the 48S pre-initiation complex. So when you have now the 43S being mounted on top of the messenger RNA, you're going to be forming the 48S pre-initiation complex. So let's go ahead and do it. And the next thing that will happen once that complex has been formed is the small Rosas unit together with the tRNA will start scanning the messenger RNA. So everything will be moving together. So factor 4G is also moving together with all of these. It'll eliminate secondary structures, and it will allow for all of this to continue scanning. And this scanning process, let's actually put it down here because it's very important to keep this in mind. This scanning process is searching for a very specific type of consensus sequence, which is the one that indicates this is the right start colon to start translation. Now, that scanning process is searching for the Cossack consensus sequence. So the Cossack consensus sequence is defined by a few nucleotides that are typically conserved. The most important nucleotides are located in the -3 position and the +4, and that's surrounding the location of the actual start codon. So the -3 and the +4 are the critical elements of the Cossack consensus sequence. The Cossack consensus is in the position -3, there is an A, then two nucleotides after that, there is the ATG, which will be, so that's in DNA language. So in RNA language, it'll be AUG. And then in the position +4, there is another G. So G in the +4, this is the +1 location. Then in the -3, there is typically an A. And then sometimes there are other sequences that also facilitate the recognition of the Cossack consensus sequence. So there is a CC that is frequently found in between the A and the AUG. Then there are other sequences further down in the upstream region from the translational start site that also seem to play a role, but they are less critical in the recognition of the start site. So the start site is the AUG codon, and remember, the messenger RNA is going to be read 3 nucleotides at a time in the process of translation. So the ribosome will be recognizing codons. Every codon is made of 3 nucleotides. So one codon is 3 nucleotides. And by the way, when you have 4 bases that are going to be read in groups of 3, the total number of combinations that you will have will be 4 to the 3, which means 4 times 4 times 4, which means it's a total of 64 different potential combinations that you can form. So you have 64 potential codons. Considering that there are only 20 amino acids that are normally present in nature, that means that our genetic code is redundant, meaning there is more than 1 codon in average for every amino acid. And there are very few exceptions for that. Perhaps the most important to mention is for methionine, which there is only one codon that codes for methionine, and that is the AUG codon. All right, so going back to this, the scanning process then is searching for that start codon, for the Cossack consensus sequence. And the whole thing, the whole complex, so including, remember that this is not just the small responsive unit, it is moving together with everything. Even this structure that is in the cap is also associated here and is moving along with all of this, because the helicase activity that is mediated by factors 4A and 4B is also important during the scanning process. So all of this is moving together with the 43S pre-initiation complex. This is all a single complex that is moving all together as the scanning process is proceeding. Now, the next stage will be when the start codon is actually recognized. And when that happens, then there is a change in the shape of the scanning complex. Oops, let's undo this. Let's copy this, then we can emphasize this. Going to be copying all of those components right here. All right, so now let's assume that the start codon has been recognized. What is going to happen at that moment when the start codon is recognized? So now you're in the Cossack consensus sequence. You have found the Cossacks. And what will happen then is that there will be a very close interaction that will happen between the tRNA and so the anti-codon region in the tRNA and the Cossack consensus sequence in the messenger RNA. And that interaction is going to produce a change in the way that the tRNA is sitting in the messenger RNA. There is a small change, a molecular change that will be sensed by the factors that are located in the immediate vicinity of the tRNA. So that will be factor 1A and factor 5. Factor 5 will be sensing that through factor 1A, and the moment that factor 5 senses that change, factor 5 will be activated by that sensing mechanism that allows it to recognize that the tRNA is now on top of the proper Cossack consensus sequence. So there is an activation event that happens that then will be passed on into factor 2. So remember, this is a GTPase. Factor 2 is a GTPase. And that sensing mechanism that is now activating factor 5 will be then passed on to activate the GTPase activity of factor 2. So factor 5 will activate the GTPase activity of factor 2. In other words, what I'm saying is that eukaryotic initiation factor 5 is the GTPase activating protein, GTPase activating protein for factor 2, for eukaryotic initiation factor 2, which means now factor 2 is active. It'll then take that GTP and it'll hydrolyze it into GDP. And when that hydrolysis takes place, there is a strong change in the shape of factor 2. And when factor 2 changes its shape, it then pushes away the other factors that are located nearby. So factor 5 and factor 2 itself will dissociate. Factor 1 will also dissociate. So this is difficult to actually show this way. So rather than trying to move factors around, what I'm going to show is what happens after all of that. So what happens at the end of all of that is, well, so I'm making a copy of everything. And what I'm going to do is delete things, which probably makes more sense. So I'm deleting factors one at a time. So now that factor 2 has hydrolyzed GTP, by the way, the stand loop will be gone because of the helicase activity of our factor 4A and 4B. So at this point, now, Keep in mind, these other factors are remaining in place, so the cap will still be loaded with those factors, which means it is ready for another initiation event. So the 5' end can then recruit another 43S pre-initiation complex so that you can restart the scanning process. But the one that has already detected the COSAC now is free pretty much of everything except for factor 1 that is still associated to the amino acid site, which means new tRNAs are still not allowed to come into the ribosome. So the next could be, for instance, glycine. A tRNA for glycine could be the next one. But it will not be able to enter yet because the amino acid site is still occupied by factor 1. And by the way, in order to create the first peptide bond of the new nascent polypeptide that is being synthesized, you need the large ribosomal subunit. And that's the next step that is absolutely required. So the large ribosomal subunit needs to first come here and associate. And that will happen at this point because there will be another factor that will now associate here, and that will be factor 5b. And factor 5b will be recruited into the small ribosomal subunit in the presence of GTP because factor 5b is another GTPase. So factor 5b is the second GTPase that we have in this process. So I'm going to write a T to indicate that this is GTP, and factor 5e will hydrolyze that GTP only once the large ribosomal subunit has associated to the small ribosomal subunit. So the moment that this binding takes place and you have now this large ribosomal subunit sitting on top of the small ribosomal subunit, when that happens, then the GTPase activity of 5e is triggered. And the moment that that GTPase activity is triggered, then that GTP gets turned into GDP. And again, 5b is another GTPase, which means the moment that it hydrolyzes GTP, it changes its shape. And at that point, the change in shape of factor 5b will dissociate factor 1. So it'll push away factor 1, therefore rendering now the ribosome fully ready for elongation. So at that stage, after all of this has happened, now you have an open A site in the ribosome, and that A site can now receive the next tRNA, which will be coming in association with an elongation factor, an eukaryotic elongation factor. And at that point, you will form the first peptide bond. This will all move together. Again, 3 nucleotides at a time. That's the way that the ribosome will continue to move along the messenger RNA. And the first peptide bond will be formed between these two. So that will allow for this tRNA to then go away, and the methionine now becomes the first amino acid that is now at the end terminus of the nascent polypeptide that is being synthesized in the ribosome. But that will require these to all be in the peptidyl side of the ribosome. Therefore, opening again the A site, so that then you can have the next tRNA coming into A site. And now we're talking of translational elongation. All right, so these are the typical events that take place during translational initiation in eukaryotic cells. I hope that this has been relatively clear. I don't know how clear it has truly been. I know that there have been a lot of drawings, a lot of things that have been moved around, but I hope that this video will be informative for all of you who want to learn about the process of translational initiation in eukaryotic cells. If you found this video informative enough and entertaining enough, remember to give it thumbs up and remember to subscribe to my channel. Thank you, and see you in a future episode of the Molecular Cell Biology YouTube channel.